Nos encontramos aquí en el tercer día de este simposio en el que estamos celebrando los 50 años de la Estación de Biología Chamela. Ha sido hasta ahora un simposio estupendo y el día de hoy va a ser igualmente bueno, con charlas realmente extraordinarias. Para empezar el día de hoy, tenemos la conferencia magistral del doctor profesor Toby Pennington, que es toda una autoridad a nivel internacional sobre bosques estacionalmente secos, especialmente en Sudamérica, pero también conoce la situación de este tipo de ecosistemas en México. Para mí es un placer especialmente grande darle la bienvenida a Toby y quiero agradecerle profundamente que nos dé esta charla. Para nosotros representa un honor, pero sobre todo un gran placer. Sin más, le voy a dar la palabra a Mark Olson para que haga la presentación. Adelante, Mark.
Muchísimas gracias, Susana. Le agradecemos mucho tus palabras porque realmente sí es un gusto tener aquí a una persona que, pues todos los que trabajamos bosques tropicales secos admiramos mucho y subraya su presencia, subraya la importancia de nuestra estación. Entonces, les voy a dar una breve introducción y luego pasamos a su charla. Entonces, el doctor Toby Pennington es investigador ahora en la Universidad de Exeter, en el Reino Unido, desde 2017. Antes fue el director del Departamento de Diversidad Tropical en el Jardín Botánico de Edimburgo, ¿no? Donde todavía mantiene su afiliación, ¿no? Y él es especialista en diversidad y biogeografía de plantas tropicales, con un énfasis en leguminosas que cualquier persona que ha caminado en un bosque tropical seco sabe que son sumamente importantes. Y sus intereses de investigación y enseñanza cubren un amplio espectro. Abarcan desde biogeografía de plantas, paleoecología, conservación, evolución. Es impresionante el rango que cubre. Tiene una amplia experiencia de investigación en los trópicos, especialmente en América Latina. Ha trabajado más de 20 años en 10 países. Y su investigación se enfoca mucho en la biodiversidad, biogeografía y ecología de biomas secos en los trópicos. Y tiene fuertes implicaciones para la conservación. Por ejemplo, cuando alguien quiere buscar información sobre bosques tropicales secos, por ejemplo, si van en YouTube, cuando yo hago la búsqueda, la primera cosa que sale son videos preciosos de divulgación que pues impulsó de manera fundamental Toby, ¿no? O sea, de los recursos principales que la gente de todo el mundo busca, cuando busca información, pues son productos que ha generado él, ¿no? Entonces ha tenido un impacto enorme, no solo en la ciencia, sino mucho más allá. Y ese impacto continúa, ¿no? A través de sus proyectos agroforestales, restauración, en selvas y sabanas, o sea, de las más amenazadas en el mundo. Entonces, es con gran admiración y estamos muy agradecidos por tener su presencia. Entonces, paso la palabra ahora al doctor Pennington. Muchas gracias, Toby.
Muchas gracias, Susana, por la invitación. Es un placer estar con ustedes en México de manera virtual hoy. I'm going to give the presentation in English. It's going to be a presentation with a very broad focus. I know the focus of your conference is on Chamela, but I'm going to talk about why the dry tropics, both savannas and dry forests, are important. That will be the first part of my talk, that will talk a lot about diversity. And most of the talk will be about diversification and biogeography. And then I'll finish up talking about the vital topic of conservation, which Mark was just mentioning. But before I start, I want to make some acknowledgements. My co-authors on this talk, there are 3 important people there who are sharing unpublished data that I'm going to show to you today. They are Moabi Fernandez from Bahia, Brazil, who is a postdoc working with me here in Exeter at the moment on legume diversity. Flavia Pezzini, who recently did a PhD with me working on the characteristic dry forest genus Sabre. Flavia's now, which is fantastic news, got a job at the Royal Botanic Garden, Edinburgh. And one of my oldest collaborators, Matt Lavin from Montana State. I'd also like to acknowledge the Dry Floor Network. All these people will be mentioned as I go through my talk. So why am I starting with a picture of a tropical rainforest? A picture that looks like it comes from a fantasy novel. It's actually a picture from a really serious scientific work, Martius's Flora Brasiliensis, the first documentation of Brazil's flora. And what the authors were trying to convey in this age of exploration back to the audience in Europe was their wonder when they saw the massive stature of tropical rainforests and their high diversity. These was the first vegetation they saw, for example, on the coast of Brazil. But that message has created a problem, and it's a problem that continues until today, because there's far more than just rainforest in the tropics. Now, you all know that, but many people don't. And what this map shows is that half of the tropics have a climate that's too seasonally dry to support those rainforests. That's where we find tropical dry forests, the subject of your symposium, and also huge, in fact, bigger areas of tropical savannas that I'm also going to talk about. And a really important point is those dry tropics matter to humanity. They're home to more than a third of the global population and include some of the poorest people on the planet that depend on those systems for the ecosystem services that they provide. So these are really important formations. I'm talking to an audience, I'm guessing mostly of Mexican scientists, student and public. You actually don't need to be told that there are dry tropical forests in the tropics, but it's really important that you realise for many ecologists, and particularly the Earth system scientists who were talking to the world in the last few weeks at the COP conference, they actually think about the world in the tropics as just having two lowland tropical biomes, forests and savannas. And the alternate states of these systems is what occupy the minds of people, for example, trying to figure out what the fate of Amazonia might be under climate change. But their forest either doesn't consider dry forest at all, or it mixes up rainforest and dry forest. And I'm going to try and explain to you why I think that that is a problem and why this picture here should actually be a triangular diagram, including tropical dry forests as well. So I'm going to talk a lot about biomes in this talk, and I've realised increasingly it's very important to be clear what I mean when I'm talking about biomes, because biome is a word that means many different things to many different biologists. But I think this definition here is one that is becoming increasingly accepted, particularly in ecology. The biome is a major vegetation formation defined by its physiognomy and ecological processes that can be characterized at a global scale, and that global part of the definition is very important. For those of you who are interested in learning more about biomes, as I say, a slippery word, a difficult concept, this review by Musina recently in New Phytologist is really worth taking a look at. And so quickly, just to be really clear, I'm going to explain to you what I think the main biomes are in the lowland tropics. The first one, where I started, another reason why perhaps it gets most attention is actually the easiest one to define. Tropical rainforests are tall forests with canopy 20-25 meters plus that are evergreen and closed. And we have that kind of height of trees, and we have those evergreen trees because they have high annual precipitation. But the key fact is that they have little to no dry season. There may be seasonality, but it's very limited. And that contrasts with the climates where we find savannas and dry forests. Take savannas first. If you note the rainfall figures here, some of you with sharp eyes will have already noticed that in some cases, their total annual rainfall can be higher than areas that support rainforest. But the key issue here is several months that receive little or no rainfall, and so therefore these areas can't support rainforest. And these savannas are not forests. As you see from these pictures, they are open formations with an important grassy understory. In the dry season, that grass dries, it cures, as we say in English, and it will then burn. Fires are a really important natural ecological factor in these savanna systems. And we know that because the plants there have adaptations to survive them, such as thick, corky bark and the ability of the plants to sprout from the roots. In the New World tropics, so particularly the main areas of these savannas in Brazil, the soils are nutrient poor and very acid, and the trees are actually evergreen. That's because they often have very deep roots that can reach underground aquifers up to 10 meters below the ground. And the subject of this meeting, the biome of the Chamela Biological Station, is of course tropical dry forest. I'll use the acronym SDTF in my slides. And as you all know, this is a forest. It's closed canopy and it has few grasses because of that closed canopy. But in the dry season, it is deciduous. That deciduousness, for me, is allowed in an ecological sense by the richer soils that allow these plants to regrow leaves on a yearly basis. And these forests don't burn, or they don't burn often in their natural state. And how do I know that? It's because they're full of succulent plants that have no fire adaptations. So those, of course, include the cacti. And any of you who don't believe me, I suggest a little experiment you could do at home tonight. If you've got a cactus in your garden or as a houseplant, is set fire to it and see how it's doing next week. It's not going to like it. It won't survive the fire. The high abundance of these succulents indicates that fire can't be an important part of the ecological system. So again, those of you interested in biomes, there's been a lot of confusion about actually, which sounds strange because it's quite a simple concept even taught in schools, but particularly in the dry tropics about what biomes are. I recommend you look at the new IUCN Global Ecosystem Typology. I'm partly biased because I've been involved in helping them develop their concepts of biomes in the dry tropics. It's actually a really useful publication even for teachers. It gives descriptions and maps globally of these biomes and their ecological drivers. So here is a slide I use very often in teaching. And if I was in the lecture hall with you that I'm sadly can't be today, I'd be asking you all, what did you first see when I brought that picture up? 90% of people put up their hands and say, oh, I saw a moth. I know there are some legume specialists in the audience like me. They might have said, oh, I see a beautiful specimen of Inga edulis in the Mata Atlantica in Brazil, which happens to be pollinated by a moth. But I'm showing you this slide for serious reasons, because the fact that many people do not see plants is receiving increasing attention in the literature under the banner of plant blindness or plant awareness disparity. And it's a real problem for conservation of plants and ecosystems globally. But I think there is an analogous problem, which is that the world is blind to some biomes. So that in English could be called biome blindness, la invisibilidad de biomes in Espanol. In this paper, we've characterized it as biome awareness disparity, which has the acronym of BAD. And biome awareness disparity is a bad thing, because the fact that many people do not see tropical dry forests and tropical savannas is creating issues. And so in this paper, which only came out a month ago, what we did was we looked at the Twitter accounts of several organizations that are involved in the United Nations Decade for Restoration, which has just started. And what we found was that they were putting out 10 times as many tweets about forests than they were about open systems such as tropical grasslands, which can't be a very good thing if those tropical dry systems are covering half of the world's tropical area. And what I want to make the case to you is that that biome awareness disparity matters because the biodiversity of tropical dry biomes is really special. And of course, everybody knows that in relation to the charismatic fauna for example, of African savannas. But what about plants? So actually counting the number of species in a global biome is difficult because global biomes span many countries. Most estimates are done by country. So here, and I'm sure you could do this for Mexico, but I'm going to give you some examples from Brazil. The Brazilian Flora Group a couple of years ago produced a very authoritative number of flowering plant species in the Brazilian Amazon. That is a big network of nearly 300 Brazilian and international taxonomists. It's the best number we have. It's a big number, nearly 12,000 species. But how many species are there in the 2 million square kilometers of the savannas of the Brazilian Cerrado? So that is a big area, but it's not nearly as big as the Brazilian Amazon. And many people are surprised to know that from the same source, what we see is more species, okay, only a little bit more, but more species than in the Brazilian Amazon, and 30% of those species are found nowhere else. So the diversity in some of these tropical dry systems is astounding. And thinking about that really matters. Here's a paper that was published in Science a couple of years ago. It received massive global publicity. It was a good news story. It's suggesting that trees are an extremely powerful way of drawing down carbon dioxide and mitigating the terrible problems we have at the moment with global warming. The map there shows in green areas where these authors were suggesting that most trees could be planted. So it turns out, I'm not a carbon scientist, it turns out that their calculations about carbon sequestrations were somewhat out. But for me, there is a fundamental problem. And that is that most of those areas in the tropics they were suggesting we should plant trees are savannas. And savannas are not forests. And if we plant trees in savannas and close the canopy in savannas, we lose much of the savanna plant diversity that can't survive shade. The afforestation in tropical savannas is really problematic, as was pointed out in a response to this paper. This to me is a direct result of people not appreciating non-rainforest biomes in the tropics. But much of my research, so to come to dry forests, so the focus of this meeting is focused on these dry forest floras and asking the question of why they're special, or are they special? And I started off by doing this more than 20 years ago by doing a really good way of doing it is by doing inventories, marking and identifying every tree in a specific area of a piece of forest. And here's my Peruvian friend and colleague Aniceto Dassa in a Peruvian interandean valley doing just that, measuring a tree of the genus Cavinilesia. And that work takes a long time. Over a period of 10 years, with many Peruvian collaborators, I did inventories in various interandean valleys, such as the Apurimac Valley on the left, the Mantaro Valley on the right. The results of this were incredibly interesting. They showed amazing endemism in some of these valleys. So the Mantaro Valley in northern Peru has 40% endemism of woody plants. To put that into context, that's more than the plant endemism on the famous Galapagos Islands. But one thing that fieldwork taught me was it was going to take an incredibly long time to get a data set of inventories that covered all of the dry forests in Latin America, because of course they're found everywhere from Mexico, right through the Caribbean, through South America to Argentina. So to address that problem, we assembled a network which is now called DryFlora, and it was a network of taxonomists and floristic botanists working in these formations across the tropics. And I know from, like, Mark told me, he posted a message in the chat, it's really nice to be able to acknowledge and thank Alfonso Delgado Salinas from UNAM for his leadership of the Mexican data set in this project. The first phase of this project was essentially taxonomic. We needed to know that we had accepted names and not synonyms in our data set. We needed to know which inventories were authoritative. And Alfonso did that, and a group of other individuals you see in this photo did that for all the other neotropical countries in which we find tropical dry forests. What we found in 1,600 inventory sites, so that sounds like a lot, But if you think about the number of countries in the area these dry forests cover, it's not that many. We found these 7,400 species of just freestanding woody plants, no herbs or lianas in our data set, unfortunately. If you look at that total number, that is really approaching that number I gave you for all angiosperms in the Brazilian Amazon. These forests have amazing diversity at continental scale that was underappreciated. That may seem puzzling to some of you, but it's an absolute fact that local diversity in tropical rainforests is much higher than a local diversity in a tropical dry forest. You can find 300 species in 1 hectare of Amazon rainforest. You don't find 300 species in 1 hectare. You might find 20 or 30 tree species. But what is causing that high number at continental scale is high floristic turnover, high beta diversity, which is what we demonstrated in this paper that was led by Karina Banda as part of her PhD in Edinburgh. What we showed was that each separate area of these tropical dry forests houses a really distinct flora, often with high levels of endemism, and it's the floristic turnover amongst those areas that gives us that high total. So I don't really need to read the conclusion on this slide to a Mexican audience. You know that tropical dry forests are important. You know that your dry forests in Mexico are the most species rich in Latin America, which doesn't obey the supposedly universal law of the latitudinal gradient of tropical diversity. But many people don't seem to know this. And that paper below, I've not shown the authors. I do not want to call anybody out. It's a very good paper about connectivity in the face of climate change of tropical forests, but they list it in their title, because if you read it, that paper's about just tropical rainforests. They don't deal with tropical dry forests at all, and their problems of connectivity may be very different. So now I want to move to the meat of my talk about the evolutionary and biogeographic history of tropical dry forests. And so I've done the diversity bit of my talk now. This bit is a bit longer and a bit complicated, and it has a winding road that is actually going to lead us to the Chamela station because it's your 50th anniversary, and so I needed to get there. And it's worth me pointing out that through these topics, talking about the age of the biome, the age of the species in that biome, biogeographic patterns, and then some thoughts about speciation, there's an increasing level of speculation. So don't believe everything I'm saying when I get to the end. I'd like to throw out some ideas for areas I think might be really interesting for future research. But how old is this biome biogeographically? That's actually a problematic question to answer from fossils, because few fossils form in dry areas. So what many workers have used are dated phylogenies. So I've done work in this area, but for Mexico in particular, and actually for tropical dry forests in general, some of the most important work has come from Susana Magallon's group in Mexico, including these papers by Hernandez about cacti and De Nova et al. About Urseraceae. And they show a Miocene origin. That's when some of the dominant lineages in these forests came into existence. And interestingly, to give you some new literature, I reviewed this paper a year ago. Just come out. There's a new macro and micro fossil evidence for what looks convincingly like a tropical dry forest, again in the Eocene in South America, in Colombia, by Camila Martinez, Carlos Jaramillo and co-workers. So it's a pretty old biome going back to the Eocene. And I've done quite a lot of work looking at the age of individual species in this biome, and that was partly serendipitous. So I'm going to talk to you a lot about legumes now. One of those legumes is Cyathostegea methusii. It's the sister genus of Atelia, which is an important genus in your Mexican dry forests. An obscure plant, but actually I happen to know quite a lot about it because it was studied by my first PhD student, Helen Ireland. It was thought to be endemic just to, if you can see my mouse, the southern Ecuador and northern Peru, but in some of those inventories I showed you in the Mantaro and Apurimac valleys, we found plants that were clearly Cyathostegia methuseae. So an interesting range disjunction. We collected leaf material and sequenced DNA. So just excuse me a minute, I'm going to turn my light on because I think it will improve the lighting in my office for me. And this is the phylogeny we got back. So note two things there. One I'll come back to is the structure there. The fact we don't see a mixing of colours. We have clear patterns of geography. But the ages of those species, we see splits between populations of the same species of 5 million years. This single species, Cyathostegia methuseae, stretches back 12 million years. So what we discussed in this paper was tropical dry forests being museums of diversity. And we then went looking for those patterns in other legumes. And the group of legumes we went looking for them were the rubinioid legumes. So I was in Matt Lavin's lab in Montana when I was working on Cyathostegia. Matt there was working on the rubinioid legumes, a bigger clade of legumes. And we find exactly the same patterns there. So it might be difficult for you to read these numbers, but the numbers on the branches are stem ages for species of Andean dry forest rubinioids, 13 million years, 10 million years. These taxonomic species are monophyletic, they're old, often with long stem lineages. But some of you may be asking yourself, well, what does he mean by old? Why is he saying these are old? How do we know how old a plant species should be? I actually don't know the answer to that question, but I'm making some comparisons with tropical rainforests. And the first study in molecular biogeography in my group was on the genus Inga. We sequenced a bunch of species from across this 300 species rich genus. And we got what really is a rather poor, rubbish phylogeny. It's unresolved. But what that lack of resolution told us was that those species must have arisen very recently. All 300 of them have arisen in the last 6 million years. Many of them have arisen during the Pleistocene, the last million year or so. They're much more recent than those dry forest patterns. And this slide that was put together by my Brazilian friend and colleague Domingos Cardoso illustrates that really nicely. So you're not going to be able to read anything here, but at the top on the right I have Inga. At the same scale I have a portion of that rubinioid legume phylogeny. Look how much deeper those patterns are in the dry forest rubinioids. So with Matt Lavin and other co-workers, I published a series of papers suggesting that dry forests have older species in them. This is where I want to show you some unpublished data to demonstrate that the pictures may be not quite so simple. This is Flavia Pezzini's target capture phylogeny using hundreds of genes on the charismatic dry forest genus Saber. Again, you don't need to read the species name, but in this South American SDTF clade, you see the great range of floral morphology. Many of those species there have arisen in the Pleistocene, in the Brazilian Caatinga. Staying in the Caatinga, this is work that's in review by Moabe Fernandez. So Moabe, this is his PhD research. He's now working on a broader legume project with me. He looked at 20 different phylogenies containing Caatinga inhabiting species, of which many were endemic. And quite a few of those showed the picture we see here in the genus Lutzelbergia, where in colour we see some Caatinga species, and we see that they have evolved very recently from non-Caatinga ancestors in black. The box plots below show when the shifts into the Caatinga happened in those dated phylogenies. And again, they go back to the Eocene, so it corroborates the old age for the biome. Arriving since then, and many of the in situ speciation events, so things that happened in the Caatinga, are really quite recent, happening in the Plio-Pleistocene. I'd like you to, I'm sure it will get accepted soon, read Moabe's paper, and the case that he makes for the Brazilian Caatinga is much of that recent speciation is driven by different geologies found in the Caatinga. We have crystalline rocks, we have sedimentary basins, and we have limestone karst areas up there in the top right. And that edaphic variability seems to be very important there. And I think this illustrates a very important point that different dry forest areas in Latin America will have different evolutionary histories because their contexts are different. But there will be some similarities, as I will come back to later. So it's a bit more complicated than maybe I thought, because we see recent diversification in Brazil's dry forests in the Caatinga. And I think the same is likely to be true in Mexico if we look in more detail, and there are hints of this in this paper by Victoria Sosa and co-workers. But I would maintain, and this is what Matt Lavin and I wrote in the 2015 paper, that we would still expect tropical dry forest species to be on average older and tropical rainforest species to be on average younger. Now that might seem counterintuitive because fossil record, and there's a better fossil record, tells us that tropical rainforests are the oldest biome. They go back right to the beginning of the Paleocene, just after the dinosaur extinction. So that might seem a contradiction, but a new model by Eric Kernan, who's a brilliant young worker in biogeography and diversification, I recommend you look at his papers. He suggested in this paper about Meliaceae that what we may be seeing is constant lineage and species turnover. So it's very interesting. If you look at the fossil record of Cenozoic Latin American rainforest, back in the Paleocene and Eocene, they had higher pollen diversity than they do today. That suggests that we must be having evolutionary radiations going on all the time and high amounts of extinction going on. So what model I have in my head is in these rainforests, different lineages have their, as we say in English, time in the sun. They diversify at different times, and then when the circumstances are not so favorable, they suffer extinction. So even if lineages are old, the whole lineage may not go extinct, many species will. So we will see recent species in recent diversifications, even if some lineages are old. And now I want to come on to reasons why dry forest species may be on average older. And it may reflect for me patterns of dispersal limitation, is how hard it is to disperse into and establish in a biome. And we get hints about this from the geographic structure of our dry forest phylogenies. So if you look in detail at the robinioid legume phylogeny, you ask the question is, what is a Mexican dry forest robinioid most closely related to? Generally the answer is it's another Mexican species. We have high geographic structure, not from Mexico, but from the interandean valleys of Peru, where we see a small clade of 4 species from Peru and adjacent Bolivia and Argentina. So that kind of geographic structure, particularly when these populations are split by mountains, makes many people think that it all must be about vicariance, for example, the uplift of the Andes. But it's fairly clear looking at these phylogenies, actually what is structuring the biogeography, and I think must be influential in diversification, is dispersal. And here's an example that illustrates that. 2 sister species in the robinioid phylogeny, Corsetia pacifica from Mexico and Corsetia ochroluca from southern Ecuador and northern Peru, split from each other 7 million years ago. That must be a disjunction explained by dispersal. There's no vicariance event that can explain that. And that dispersal paradigm is underlined by this excellent paper from Chuck Davis's group by Charles Willis, which shows that the Malpighiaceae in your dry forests in Mexico are the result of 33 dispersals from South America, beginning in the Eocene and continuing till recently. So dispersal is what is driving the biogeography, and I think influential in diversification. And now I want to do another comparison with rainforests, because it helps think about speciation processes. So in a study that I undertook principally with my collaborator friend Kyle Dexter, we looked at 4 really important Amazonian tree genera. They're abundant and species rich. One of those was Inga. And to demonstrate our sampling, we're collaborating with ecologists who sampled all the Inga species, which can be up to 60 in one local population, for example, in Ecuador. So in this diagram, for example, in blue, you see all the species from southern Peru. What you see in that phylogeny is no geographical structure. It's a kaleidoscope. It doesn't look like those seasonally dry tropical forest phylogenies. These local communities, it looks like they have been assembled by dispersal. And the way Kyle looked at it more asked about the phylogenetic diversity, the lineage diversity in each one of those communities. And what we see in these diagrams for each genus, for those different communities, that the phylogenetic diversity is effectively random. They are a random draw by dispersal from the regional meta-community. That tells me that local Amazonian tree communities are assembled by rampant dispersal, higher levels of dispersal than we must see in tropical dry forests. And that high alpha diversity we see in forests like the one in this photo does not reflect in situ regional speciation. For me, it's much more likely to reflect a speciation process that involves dispersal. And this, thank you for your patience, is leading me on the road to the Chimela Biological Station to give an example of how I think that speciation is happening from a legume, Andira, which was the subject of my PhD. And I'm showing you here a species, Andira inermis, the most widespread in the genus. It's remarkable. It's found all over South and Central America, and it even reaches Africa, and that's a natural distribution. In my monograph of Andira, I pointed out that Andira inermis looked very like two other species, one called Andira multistipula that grows in flooded forests in the Western Amazon, and one, and this is where I finally get to the Chimela Biological Station because it grows there, is a new species that I described called Andira jaliscensis from Jalisco, which is endemic to those northern Mexican forests. And it's like Andira inermis, except for it's covered in hairs. It's quite distinct. And what I suggested in that monograph was that Andira inermis might well be the ancestor of these two other species. And I suggested that that might reflect a peripatric speciation mechanism driven by dispersal. So this was long before I saw any phylogenies. And I won't pretend this was any great idea of mine. If you read carefully some of the great 20th and 21st century taxonomists such as Alwyn Gentry and Ian Prance, they talk a lot about widespread species being the progenitors of other species. But a master student in Edinburgh did do a phylogeny of Andira. That's part of a publication which actually is not about Andira. That publication is about savanna evolution. I don't have time to talk about it. The phylogeny in detail is buried in there. And let me show you a detail from that phylogeny. And it fits the prediction perfectly. If Andira inermis was the ancestor of multistipula and jaliscensis, we'd expect those species to be nested within it. That Andira inermis would be non-monophyletic, the ancestor. And that would be the case, at least if the speciation was very recent. And that is exactly what we see. And we see that Andira jaliscensis must have a very recent origin in northern Mexico from Andira inermis. So that, as I say, is a somewhat speculative suggestion about how some speciation processes may be happening in both tropical rainforests and tropical dry forest plants driven by dispersal. But I think there may be more elevated speciation in tropical rainforests because that dispersal is happening more frequently. But I now want to go back to dry forests. Still thinking about dispersal, make the point that we often see in tropical dry forests that dispersal happening amongst the disjunct areas of that same dry forest biome. And what that suggests is the phenomenon of phylogenetic biome conservatism, that these lineages are tending to stay in their same ecological setting. And two papers from Colin Hughes' group in Zurich have demonstrated this very nicely. One's about the legume Caesalpinia clade, and another is more general. I'll describe both of them in turn. First, the Caesalpinia by Edeline Gagnon. And on the left, we see the biome mapped on that phylogeny. It doesn't matter, you can't even read the genera. But the red colour is the succulent biome, so more or less that's tropical dry forest, transcontinentally in Africa and in the world. And what we see is the red is clustered in that phylogeny. Again, just contrast that to the rainforest pattern that I showed you. And the fact that it's all clustered in that biome means that we have not seen many biome switches, which is exactly what Edeline's conclusion is. That the number of biome shifts observed in their phylogeny is always significantly lower than tests where they look at shifts obtained with randomized tip states. And taking that forward to a group of 10 phylogenies, Jens Rinkelberg showed, for what he's calling the succulent biome, you can see mapped there, exactly the same conclusion, that we basically see most of these clades are confined to that biome. And the age of the disjunction suggests that these lineages are hopping between different areas by dispersal. But those, even though those phylogenies, particularly Jens Rinkelberg's study, is in advance, they're individual phylogenies. They're suggesting biome conservatism. Is that true for these fantastically diverse floras that I told you about at the beginning of my talk? I think that is true. That is demonstrated by a study published just last year by Ricardo Segovia, who's was a postdoc in Edinburgh working in Carl Dexter's group. And what Kyle and Ricardo did was generate an absolutely enormous, fantastic data set of tree inventories from right across the Americas, both temperate and tropical. What you see there on the left is an ordination in evolutionary lineage space. What you see, and that's maybe not a surprise, is that the non-tropical regions separate out first in red and in blue. The tropical areas are the other side of the ordination, but they split along a precipitation line. They split along a availability line. We see the rainforests in one group in green and the tropical dry biomes in another. And that pattern wouldn't emerge if we weren't seeing biome conservatism, in other words, certain lineages being confined to certain biomes. And this, I enjoyed putting this talk together this week because it made me think about where I think next steps should be. And something that's been interesting me for many years is thinking about what are the evolutionary barriers that are structuring our phylogenies. What are they, and what are the changes that are needed to allow lineages to smash through them? And I chose my verb very carefully there because that phylogenetic biome conservatism for tropical dry forests, it suggests to me it's a really difficult biome to survive in. And I think for woody plants, if the driver in the tropics is water availability, surely this must be about hydraulics. And that's something I've been able to think about in recent years in Edinburgh because my colleague Lucy Rowland, she's in the office next to me, she works on the hydraulics of tropical trees. These sections here are from a postdoc working with Lucy Jehovah Lourenço, taken from wet forest in coastal Brazil and very close to the adjacent dry forest. Different species, but showing very different wood anatomy. One with big xylem vessels surrounded by parenchyma, other with much more smaller xylem vessels surrounded by fibers. Is it these kind of changes are the ones that are governing things? I think it probably is. One hint, it's a paper published just last year by Pablo Sanchez Martinez looking at phylogenetic signal in some of these plant hydraulic traits and suggesting they have high levels of phylogenetic signal. It means they're not changing that much. So this was a study which actually was poorly sampled right across angiosperms. The authors would be the first to admit that. But it really looks to me consistent with this picture of this being a very important and difficult to evolve trait. What do we need to do? I think we need to do, and it was very nice to be able to chat a little to Mark Olson before this talk, we need to do studies just like Mark is doing, careful anatomical studies of plants, of woody plants in different biomes to understand how their hydraulic systems differ. We don't have enough of that basic information from these tropical dry biomes. And I could suggest to you some possible case studies, and some of them could be done at the Chamela Biological Station. So back to Andira inermis and its relatives. So Andira inermis is a fascinating species. My picture there, and it is one of mine, is from Amazonian Peru. That's a very wet area. Andira inermis grows in very wet areas in South America. Oddly, when it gets to Central America, it is in dry forests. When I was doing the taxonomy of Andira inermis, I thought that I might end up splitting it into two species, but I could find no macrophylogenetic differences in thousands of sheets. But interestingly, what this phylogeny gives us is a clear phylogenetic framework for a species that grows in flooded forests, which has arisen from a species growing in rainforests. And it looks like Andira haliscensis has arisen from Central American ancestors.
Toby comentó que tiene una conexión que puede ir y venir. Mientras esperamos que regrese, les invito que vayan colocando sus preguntas, por favor, en el chat y podemos abordar sus dudas y comentarios cuando él puede regresar. Entonces, agradezco su paciencia mientras esperamos que se pueda volver a conectar. Y creo que como yo, seguro están disfrutando mucho ver que alguien enfatice de una manera tan contundente y con tan buenos ejemplos, puntos que nosotros que trabajamos bosques tropicales caducifolios, siempre encontramos retos o problemas para convencer a las personas que trabajan en la zona templada o en bosques húmedos, ¿no? Por ejemplo, la gran extensión geográfica de estos ambientes, ¿no? Muchas veces la gente no se da cuenta que la gran mayoría de los ambientes tropicales, por debajo de los 1500 metros, pues son ambientes estacionales, no son selva. También la importancia ecológica para humanos, la extensión en estos ambos sentidos, o sea, que sus suelos son muy buenos, ¿no? Entonces son mucho más importantes, tienen densidades poblacionales mucho mayores que selvas altas, ¿no? Entonces tiene una gran importancia para humanos. Tantas plantas, por ejemplo, aquí en México, frijoles, maíz, fueron domesticados de estos ambientes tropicales secos, ¿no? No de las selvas, a pesar de la gran diversidad de especies en las selvas. Todo esto ha enfatizado aquí Toby y de una manera muy contundente. Entonces, pues ya regresó y entonces vamos a volver a su charla.
I had a complete system crash. I've got no idea why. So I'll just reopen my presentation again. I had to restart my computer. I'm very sorry, everybody, about that.
Ningún problema. Muchas gracias, Toby. Estábamos hablando de que estamos muy agradecidos por los temas que estás enfatizando, la extensión de los bosques,
sí.
su importancia fascinante. Muchas gracias.
Estoy casi terminando. Voy al final.
Muchas gracias. Veo que están empezando a llegar sus preguntas. Muy interesante lo que estaba comentando Toby, que no se queman, y esa prueba que sugirió de tratar de quemar un cacto, muchas veces es difícil convencer a personas que no trabajan en estos ambientes que no se queman. Entonces, vemos que ha pensado mucho en esto también Toby. Agradecemos mucho que haya hablado de este tema también. Ya, pues vamos a regresar a su plática y muchas gracias.
So, Mark, I think you're not seeing it in presenter mode at the moment, right?
Not yet.
How's about now?
Not yet. If you want, I can remove this and you can select it again if you like, which worked the first time.
Yeah, let me see if I can get it back again.
You bet. Great. So we're seeing your slides again.
You see my slides again? Yeah, oops. Yeah, I don't know why, because I've not got two windows. I'm struggling to do what we did before.
We can go with this. There we go.
Yeah, I think maybe we should go with that just to keep going, because otherwise I think we could be a while. So are you seeing my screen now?
Looks great, yeah.
The slide? Okay.
Sure enough.
Yeah, so yeah, apologies everybody for the technical problems. So where I was saying that we've got a really good study system here, where we could look at the evolution of some species that are occupying very different biomes, where we know their relationships really, really carefully. And I think to come on to my next slide is a good way of looking at that would be to take some genomic approaches. So this, I'd like to thank Prof seeing about this tolerance in Arabidopsis. So they change the size of the pits of the xylem vessels, which is very important throughout. We target capture approaches such as the ones that I mentioned that Flavia Pizzini used. We could put those genes in our bait sets and pull them out of non-model species and sequence them and find out their sequence differences and look at patterns of selection in some of these clades where we have a phylogenetic framework understanding what species have evolved from each other. And we can do this if we look also at a comparative transcriptomic approach. So here, actually, we don't even need any candidate gene information. I think it could be very interesting, given that the cambium is giving rise to those different xylem tissues, is we could do transcriptomic sequencing. We could do RNA-seq from the cambium of sister species found in different biomes. In other words, from cambium samples taken at the Chamella Biological Station from Andira haliscensis and Andira inermis. And finally, and I haven't had time to mention this, in the genus Inga, I'm doing quite a lot of work using genome sequencing and assemblies. If you have a reference genome, that's a good reference genome, you can do some less deep sequencing of non-model species and get a very long way in terms of understanding the genetic control of some of these. One possible place to do it is not tropical. An obvious place to start would be poplar, which is the tree model organism. So yeah, again, sorry for the technical problems because I was almost at the end. You know, my concluding thoughts scientifically are these, which is that rainforests are very different from tropical dry forests and from tropical savannas, which I haven't mentioned much. They're different in their floristic composition, they're different in their patterns of floristic turnover, and they're different in the evolutionary history of their component floras. And those evolutionary differences lead me to think about these biomes as evolutionary theatres. We see different evolutionary and biogeographic patterns in tropical rainforest, tropical dry forest, and tropical savannas. And so it's worth thinking once again to emphasize that we can't think about the tropics in just one biome bin. It's not all rainforest. We can get a lot from studying tropical dry forests and tropical savannas and comparing processes in each of those biomes. So I want to finish off with a few thoughts on conservation. And my next slide is not going to work very well. It was animated, and the points that it makes are the fact that Amazonia, despite the fact that we see so much in the news about it, actually is one of the world's last wildernesses. It remains 80% intact. And that's a piece of good news. I'm not saying that we should be complacent, but it's worth considering how that compares with savannas and tropical dry forests. So the Brazilian cerrados, where I'm working a lot at the moment, 50% of that area, so that is 1 million square kilometres, have disappeared in the time that you have been working in the Chimela Biological Station. It's disappeared to intensive cattle and soy agriculture. But as I think all of you will know, those tropical dry forests, that the ones we hear least about, are perhaps the world's most endangered tropical forests. There's less than 10% remaining in many countries. And if we look at how well protected those tropical biomes are, they are underprotected. So this is something else that Dryfloor did in our paper in Science. Again, there's some figures for Brazil. I'd be really interested to know what the figures are for Mexico. In Brazil, Amazonia does pretty well. 30% of it is protected if we include indigenous reserves. That's more than the HE international target of 17%. But if we look at Caatinga and Cerrado, so dry forests and savannas, only about 8% of those are protected. And in the dry forest, if we just look at national parks, so fully protected area, only 1% of the Caatinga, which is full of endemic species, is under protection. So compared to rainforests, our dry forests are really badly underprotected, and that neglects the fact that many areas, so I'm highlighting ones in Peru here, are endemic and species rich but lack sufficient protected areas. So I was delighted that a couple of years ago the first reserve was declared in the Marañón Valley, Peru. It's amazing it took that long given how important that area is globally in terms of its endemism. And it's not just species endemism, they harbour unique lineages as well. And so my final slide, and this is a slide that I give to lots of people in lots of different places after these kind of talks, is to remember that these forgotten forests, the forgotten dry forests in particular, and the savannas of the tropics. But I think you as a Mexican audience know that. And you know that because you've been researching in Chimela for 50 years. And so I think in some ways, Mexico is a beacon for the world in to focus on tropical dry forests. And that's my message to you, is you need to be proud of that and to put that message out. And so I'd like to thank you very much for listening. Apologies again for the technical issues. And I would like just to finish with thanks to the fantastic Dryfloor network. It now numbers more than 100 people. And as Mark mentioned, if you want to look at our dry forest film, just put that into Google, or you will find it on the Dryfloor website. So thanks very much.
Muchísimas gracias, Toby. Me gustaría pasar a las preguntas. Tenemos un poquito de tiempo, entonces vamos a empezar con Jordan. ¿Tú estás viendo, Toby, la pregunta?
Yes, I am. That's a really good question, and so yeah, thanks very much. Thanks very much for that question. As I say, I've been thinking about some of these issues. So yeah, of course, you know, the evolution of the CAM photosynthetic pathway is going to be very important for those succulent cacti. And of course, stomatal densities, pubescence, are also going to be really important because the leaves are a key part of the hydraulic system. I mean, I actually suspect that Mark may be better qualified to answer this question than I am. But I think, you know, we were commenting earlier, what I think strikes me is I was looking at some of that developmental genetic literature with some help for the first time this week. And what strikes me is the lack of fundamental anatomical information that we still have. And so I think there's a massive value in that kind of work. And so whilst I'm suggesting some genetics might be really important, I think it needs to build on some important comparative biology before we go there, or potentially at the same time.
Puntos súper importantes.
Yeah, so again, that's a very good question, Julieta, about fire in tropical dry forests, and I think that this stems from misunderstandings. And I'll bring in savannas here rather than just dry forests. So I think there's a perception that tropical dry forests do burn quite a lot, because when there are humans around in the dry season, it's very easy for them to set on fire. So I think there's a perception that fire is a natural part of that system. Infrequent fires possibly were. I don't think we've got a very good handle on the fires. So again, that's another research area for any paleoecologists in the audience. I don't know of a really good study that's looked at fire history in tropical dry forests. There are a lot of people working on that in the Amazon, and I think that needs to be done. But flipping to savannas, there's almost the opposite problem, where because of this obsession with forests, in Brazil, there's been a lot of programs to control fire in cerrados, and what that does is results in a massive cut in the species diversity. We lose a lot of the herbaceous and the subshrub flora. So understanding these ecological drivers, that's why I was highlighting that IUCN ecosystem classification, I think is fundamentally important at quite a basic level. It's something we really should be thinking about teaching all our students, because it's central to management and conservation.
Muchas gracias, Toby. Sí, es un punto muy interesante de venta difícil. Yo tendría una pregunta. Any idea what percentage of the lowland tropics are seasonally dry?
No, I don't, and I'm actually, I didn't talk about it today, but I'm lucky to be part of quite a big new project looking at carbon balance across the dry tropics globally, which has got partly a remote sensing focus, or very largely, but also a large ground-based focus that's being led by Kyle Dexter from Edinburgh, is working right across Africa and the New World. And I think that some of that remote sensing data is going to be important to answer your question, Mark. And so it's a really good question. So I think, you know, that of course, having given a talk sort of making look like those biomes are in neat boxes, of course they're not in neat boxes. These areas of transition are really important. The area of transition, like between dry forest and selva, is really important, but it's more than about rainfall. So I'll give an example. So I think it's back to water availability and hydraulics. I've worked in Tarapoto, Peru, a poorly known area of dry forest that receives, I think, 800 millimeters. It's got quite a cool dry forest. The fieldwork there was horrendous. It was very wet. It was a clay soil. It was steep slopes. We were slipping around everywhere. I think that clay soil has got much better water retention properties. So the water that the plants perceive is very different because of the soil. You know, you can have 800 millimeters on a different soil, a more sandy soil, that is going to, you're going to get an extremely open, scrubby system that doesn't look like a forest at all. So again, I think there's a lot of work that needs to be done here, looking at soil science and water availability. But I hope that this big project is going to help map and some of our understanding. And just as an addition, actually, at the drier end, I see some differences between what people are calling the succulent biome, so the drier end, which aren't really forests, and what I've called tropical dry forests. So just to confuse everyone, to be really clear, for me, tropical dry forest is I'm including everything from those cactus-rich scrublands to quite tall dry forest. A lot of people don't like that. I keep them together because I don't know where to draw the line. But one thing I'm really interested in, is there a line there? And I think there might be a phylogenetic diversity line. I think there might be something really different about those dry formations. So sort of having given you a really clear picture of those biomes, I see it as a very open area of research where we draw some of those lines. So that's a very good question and probably a very bad answer, because I guess the answer is I just don't know.
No, but that point of pooling the habitats, that's a super important point. Thank you for that, Toby. We have a question from Alfonso Delgado, our Legume colleague. He asks, should we take care of herbs and shrubs as well, not just trees in our reforestation?
Gracias, Alfonso. So, absolutely. So, I'd use the word here, restoration as well as reforestation. So I'm starting to do some work again with my colleague Lucy Rowland, looking at habitat restoration in the Brazilian Cerrado that really needs it. Again, you missed some of my photos, but the sort of scale in there for intensive agriculture is something we don't see in tropical dry forests. But we really don't understand much about how the trees grow there. We understand even less about how the herbs and shrubs grow on those aluminium-rich, really poor acid soils, and most of that Cerrado diversity is in those life forms. And I think, Alfonso, as you know, in dry forests, in dry floor we produce those impressive species numbers just for woody plants. Where I really want to take dry floor next is to herbs and to small shrubs, because that's going to push the numbers even higher. There are lots in there. I mean, as all of you will know, when you get a beautiful, really rainy season in those forests, what you find on the ground, I mean, it's absolutely fantastic. I was in the Brazilian Caatinga in 2018. At the end of a drought, they had substantial rains. It was absolutely beautiful. The carpets and meadows of flowers. So my dream, and I was talking to Karina Banda about it, is we've got a phase 2 of dry floor, which is entering abundance data from plots. Phase 3, I'd really like to find money and new partners to do would be to expand our data set, take on herbs and shrubs, so we can do precisely what you're saying. But we really need to include those in the restoration and reforestation program. They're a vital part of the ecosystem, not just their sheer species numbers, but the roles they're playing in the system, the pollinators, etc.
Oh, súper importante esa pregunta también. Toby, you've got a question here from our jefa of the Chamela station. She wasn't able to get the chat in. Oh, there she goes. So she's asking about this.
So that's a really good question, Catherine, and also it's a really tough one to answer. So I think about dispersal on 2 really different timescales. So on ecological timescales, and I guess that's what you ecologists can study at the Chamela Biological Station directly. For woody plants, at least, the work tells us that there's a greater preponderance of wind dispersal in those systems, because I think they're open in the dry in rainforest systems. So wind dispersal is more important. But for the historical biogeographic angle and what's driving diversification, we're talking about rare random dispersal events, and it's really hard to say what is driving those. It looks, our phylogenies are telling us that they must have happened. And actually, it's an area of research that I think needs to be really important, is to ask the question at those historical timescale, where are we seeing most dispersal events? I'll give an example. It might seem really unlikely, like Malpighiaceae, they are, well, a lot of them are wind dispersed, right? They seem to have reached Mexico from South America very effectively, but is something that's vertebrate dispersed showing, different patterns of geographic structure? Aware of many studies looking at that in sort of deep phylogenetic time, and I think it will be interesting at various spatial scales, but I think it's really important to understand that, sadly, it's quite hard to draw conclusions, I think, about historical patterns of dispersal over a million years timescales from what are very valuable studies of dispersal for thinking, for example, about habitat connectivity in times of global change that are going on at the moment. Is those 2 things aren't necessarily going to kind of logically join together.
Yeah, that is a great question. I'd like to use the last minute we have, Toby, to ask you about what is your view of sort of plant life form diversity in dry forests? We often say, and I repeat this all the time, that these are the habitats that have the widest range of life forms, and certainly there are life forms like bottle trees that are restricted to them. Do you think there's truth to that?
And so why I love giving talks like this is you've actually given me a thought, which is it's so obvious. I've never seen it before, but I think you're absolutely right, because in those tropical dry forests, you have that succulent strategy, right? You have bottle trees like the sabres, you have the cacti. You have things that are kind of halfway there, like Cavernolaceas aren't quite as bottle-treed as sabres. You have things that look like kind of normal trunks. Then in those drier forests, something that happens, and I think it happens naturally, is you get a lot of multi-stemmed trees. I think that those could be really long-lived individuals that are able to lose whole trunks when they die, but the whole individual doesn't die. So this is one reason I haven't mentioned it. The projects I've been doing in Brazil, one thing I've been pushing with dry flora is better long-term monitoring in tropical dry forest, like spread over big spatial scales. So Chamela again is a shining example. You've been there for 50 years, but it's really important to do this over the whole geographic scale of those biomes. So in the Caatinga, when I was putting in plots, I was fascinated by these multiply stemmed trees that I think are being pruned by droughts. It's a kind of growth form you might see in the tropical savanna, but they're being pruned by fires. And so I think you're absolutely smart. We did some work in this project in Brazil. It was quite a well-funded project. We used ground penetrating radar and then actually excavated a 10-meter by 10-meter plot of Caatinga, and some of the subterranean root systems we found incredible. So maybe I should give another seminar showing those pictures. I had no idea so much was going on below ground. It's something that I imagined was more in relation to the Cerrado. And so I think you're absolutely right. And I'll go back to my basic point that I think that there's so much room for fundamental comparative biological work. So the kind of anatomical work you're doing, down to actually, if you're able to do it, is actually digging up plants and seeing what's going on below ground, because actually the ground penetrating radar didn't work very well, if I'm honest. A shovel was much better. We need to know what those strategies are, what the preponderance of those strategies are. In other words, which are the best ones? What are the evolutionary stable states? There seem to be all sorts of ways of doing things. Throw in nodulation. Most of the legumes in tropical dry forest nodulate. Why? More of them nodulate there. So this is unpublished data that I have than they do in tropical rainforests. It's not about poverty of soils. Those dry forests actually have better soils. Again, what's going on there? I've got some ideas. I think it's about needing to be able to grow really fast as soon as it rains. But I think, again, it just underlines the need to understand these forests still in the 21st century at quite a fundamental level. So it's something within reach of everybody, of the simplest student project is what I think makes it quite exciting.
Toby, thank you so much. We've clearly our discussion could go on and on here. We're going to wrap up now, but I just want you to imagine the applause of all of the viewers who are with us right now.
Thank you very much, Mark. And I'd like to apologize again, because that was not your fault. Which hardly ever happens. My computer crashed completely. Got a scary error called a blue screen. I don't know what anyone's ever had that. The only good news is sometimes it takes me half an hour to restart it because it has to check the whole disk. But anyway, apologies for that. I am really, really sorry.
No worries at all. And thank you so much for helping us to remember these forgotten forests.
Yeah, no, again, thank you for all the work you're doing and for a fantastic meeting.
Thank you.